present their presentation. Maybe you can mention your name and institution, and if you want to mention your advisor, it's okay. The control is extremely simple. If you hold it this way with the yellow on the bottom, there's a right arrow and left arrow. Push the right arrow and the slide or the next animation will advance. Just above it is a laser pointer. Okay, so right and laser pointer, all you have to know. The other thing is each one will get 4 minutes. Rick is the timekeeper. Unfortunately, the timer is very quiet. So Rick will stand up when you have 3 minutes. That means you have 1 minute to wind down. He may give a symbol when it's 30 seconds left, but in 4 minutes you have to stop or we will not stay on time. This is the one part. It's to give people an idea who everyone is. For this set of presentations, there's going to be 2 prizes from molecular dimensions. James Gordon is up here. Tomorrow he will have in the Lopati Conference Center from molecular dimensions, you'll see the prizes. It's going to be a glass cube of a PDB protein of your choice. Even if it's not a structure you determined, you will tell him and they'll work out that there'll be a picture before they engrave it in glass and it will be shipped to you beautifully. It will take a few weeks afterwards, but we will choose. And the judges are Alberto and Osnat sitting in the front row. So look at them when you speak. But they have your presentations. So we're going to start, and I think I won't even introduce you. Will introduce yourselves. So be in order. And if someone is not here, they won't speak. So we're going to start with puja, and after puja, the next person just to get ready. But then you'll look at yourselves. Just get on ready so that we don't waste a lot of time. And all the slides will go in order. So puja, please, you can set the tone of the whole session, okay? So this is that and that, okay? So don't make you triple this, right?
Thank you.
Yes, 1 second. Something's wrong? What happened?
Low battery.
Low battery to show me.
Oh, it's really bad. How could that be? Moment.
Do you have a power supply or yours?
I have a power supply. 1 second. Better?
Not yet.
It's not plugged in. Better?
No.
I have this. It's right here.
Yeah, now it's better.
Say, thank you. I didn't. It wasn't plugged in. It's a USB-C. You understand? All right. So, there's one other thing I had to say, but I don't remember what it was. Later on. We'll do that later on. Please.
Thank you. Can I start it? Yes. Good afternoon, everyone. I am Pooja Vadhwa from Dr. Bhupesh Taneja Lab, CSIR IGIB India.
Speak into the microphone.
So, the project of our lab is investigating RNA metabolism in mycobacterial species. So, coming to RNA metabolism in a cell. So, RNA metabolism involves synthesis and the degradation of RNA molecules to maintain the nucleotide pool of a cell. So, when we look into the RNA degradation of basically mRNA, so it involves the role of an ribonucleases. So, there are 2 types of ribonucleases, endoribonucleases and exoribonucleases. So, the degradation pathway is initiated by the enzymes that are RNase EG, RNase J and RNase 3. They have their specificity towards the RNA molecules. After the action of these endonucleases, then there are the intermediate products that are formed, and these are degraded by the different set of exoribonucleases that are PNPases, RNase 2 and the RNase PH. So when these enzymes act on these molecules, then the last product that have been formed are of oligoribonucleotides or the nanoribonucleotides. These are of different line, like they can vary from 2 mers to 5 mers. And these oligoribonucleotides are not only generated from the degradation of these mRNAs or the RNAs molecule, but they are also produced in the process of like cyclic dinucleotide molecules degradation pathway. So these elimination of these nucleotides is very important for a cell. Otherwise, these can lead to the misprime initiation, and that can be lethal to a cell, as this can lead to the alteration in the gene expression. So when we look into the enzyme that are involved in the degradation of these nucleotides, we found that there are two set of enzymes, that is oligoribonuclease and the nanoribonuclease. So when we look into the distribution of these these ribonucleases, there is an interesting aspect that the oligoribonuclease is present in many sets of bacteria, like in protobacteria, where it is found to be essential for a cell, whereas the nanoribonuclease is present in firmicutes, like in bacillus. And in these organisms, like in bacillus, the oligoribonuclease is absent, and the nanoribonuclease is found non-essential for a cell. Whereas the actinobacteria that comprises like mycobacteria, in that phylum of bacteria, there are these two ribonucleases are present. It means for a same set, for one pathway, that is the elimination of these small nucleotides, there are two different sets of enzymes. So we are curious to understand why the mycobacteria is possessing. Okay. Is possessing these two different sets of enzymes, whereas the one might be essential, one may be not. So in our lab, we are able to characterize the nanoribonuclease, which belongs to a DHS family of phosphodiesterases, which comprises of N-term DHS domain and the C-term of DHS associated domain. So when we characterize the structure of this protein, we found that it is having a C-term DHS domain and the N-term DHS domain and the C-term DHH associated domain linked by a linker region. And there is the C-term is involved in the activity of the enzyme, whereas the C-term assists in the binding of the substrate to the protein. When we look into the packing of this enzyme, we found that the two subunits of a dimer is associated through a linker region where it forms a hydrophobic interaction, whereas in the other proteins of the like phosphodiesterases, the hydrophobic interface is formed by the subunits like N-term and the C-term. They forms the hydrophobic interaction, which means in the NRN, the active site is like in two subunits, the active site is posed perpendicular to one another, and the enzyme is having two independent active sites. So right now, I am working on oligoribonuclease, which. So can I say 1-2 lines? Okay. So
30 seconds extra.
Okay. So oligoribonuclease is comprised of DE-DHH residues, and we are doing the structural characterization and functional characterization of this protein and mycobacteria smegmatis. Thank you.
Good afternoon, everyone. I am Shriparna Bishwas from Indian Institute of Science, working under Professor K. Sugna, and I have titled my talk as Bacterial Small Heat Shock Protein Structural and Functional Aspects. Coming to small heat shock proteins, these are a class of molecular chaperones which are ATP independent. This binds to unfolded or misfolded proteins and prevents the further aggregation. Later, they transport the unfolded or misfolded protein to ATP dependent chaperones for either refolding it back to its native conformation or further degradation. Why the terminology small? It is called small because the molecular weight is on an average 20 kilodalton. On an average, it is from 14 to 48 keda molecular weight. The basic unit is a dimer, but this small heat shock proteins form a huge oligomer as a storage vesicle. When when the condition arises, when stress comes, it disintegrates into smaller oligomers and binds to various substrates. Hence, they are highly dynamic and forms large oligomers under stress and undergo drastic rearrangement dissociation in order to execute the chaperone activity in protecting substrates. This interaction prevents the substrate proteins from aggregating irreversibly. These proteins are ubiquitously present in all kingdoms of organism. In archaea and bacteria, only 1 or 2 small heat shock proteins are present, whereas large number of small heat shock proteins are present in eukaryotes. In humans, 10 small heat shock proteins are present. Our lab focuses on various small heat shock protein from various organisms, but I work on bacterial small heat shock protein from 2 different organisms, Staphylococcus aureus and Bacillus cereus. In small heat shock protein in bacteria helps in protecting cell membrane components, bacterial dormancy, and various recombinant protein production. My small heat shock protein from Staphylococcus aureus forms a huge oligomer. In analytical, I have seen a 48 mer, and the Bacillus cereus has 2 small heat shock protein. One of them forms a 36 mer, and both show a chaperonic activity up to 50 degrees centigrade. The chaperonic activity was proven with the enzyme NDE1 by thermal denaturation and also by lysozyme by chemical denaturation. Thus, I have crystallized the small heat shock protein from Staphylococcus aureus in IFO symmetry, and molecular replacement trials are still going on. Thank you.
Good afternoon. I'm Nicholas. I'm from the University of Groningen, a group of Professor Dick Janssen, biotechnology, and my project concerns the validation of computational methods using X-ray crystallography. So in our lab, since random mutagenesis and rational design are kind of expensive in terms of time and money, we're focusing on computational enzyme redesign. Basically, they made 2 workflows, one called Fresco that improves thermostability and the other one called Casco that improves enantioselectivity. They start from crystal structures on the PDB of some enzymes of interest. They do in silico design and screening by visual inspection, and then those mutants are ranked by MD simulations. The ones that are positive to those to the screening and the simulations are tested in the lab. If the mutations appear to be helpful, then they get combined and tested again. So this is where I step in. I select the outliers, so the mutants that present a higher or opposite enantioselectivity for the Casco workflow, but also I choose one for the Fresco as a thermostable mutant. Then I try to express, purify, and crystallize these selected designs, and then try to obtain crystals, which is not as straightforward as may be as the predictions. Once I will get the structures, and I hope I will compare the experimental structures to the predicted ones. That's for improving the workflow, because we can compare the quality of the predictions, and to check also if there are any structural changes occurring in the crystal, and also to apply the insights that we will get from the structures back to the computational workflow to improve it and try to make it a bit more enzyme-wide instead for specific ones. So, thank you. I was super brief. Any questions? There's still time, I hope.
So, you have no structures yet?
Sorry?
You have no structures yet?
I have data. I'm working on those structures, but I'm kind of newbie, so it's taking a bit of time.
What kind of enzymes are you working
On dehalogenase, alkyl dehalogenase, and on aminotransferase. And the dehalogenase is the one that it's kind of hard to crystallize and solve.
on? Maybe now the next one.
Yeah.
Thank you. Forward.
Okay, my name is Adas. I represent the lab of Ron Diskin from the Department of Structural Biology here in the Weizmann Institute. Our work interest is viral host cell interactions. We are specifically interested in arenaviruses, which are prevalent in the Africa and South Americas. Those are hemorrhagic fever viruses, which are zoonotic and easily transmitted from rodents to humans. They have limited therapy, and therefore they can be used as bioterrorism. And so this is the interest of studying them. The binding subunit is the GP1 from a bigger complex of a glycoprotein complex, which is the GP2 is on the membrane, the GP1 is on the surface, and this is what binds actually to the receptor on the human host. We were able to solve a few structures, a few GP glycoprotein structures. One is the very pathogenic Lassa virus. The second was the Muruguru virus, which is non-pathogenic, and actually by solving the Muruguru virus, we by comparing to the Lassa virus, we could actually detect the exact binding site and the exact amino acids which are important for the binding site to the human host. And we also solved the non-pathogenic white water rotavirus, and recently we solved another 3 structures, Machupu, Sabia, and Guanarito, pathogenic viruses which are not published yet. Now we are trying to solve the structure of the Luju virus. And the Luju virus is interesting because it's they are subdivided to Old World and New World, and it has the characteristics of the Luju virus is not Old World and not New World. So this is very interesting to study, and we actually expressed and co-purified the Luju virus GP1 with the NRP2 binding receptor, and we could crystallize them. We got porcupine-looking crystals, which actually didn't look so promising, but we actually could optimize them a little bit and get 2.9 angstrom resolution, and I hope to improve it a little bit in this workshop. Thanks.
Good afternoon. This is Venkatreddy Dadareddy from Department of Physics, Indian Institute of Science. I'm working with Professor Ram Kumar. So we are interested in structural and functional characterization of putative protein acetyltransferase from Helicobacter pylori. So acetylation is one of the second predominant post-translation modification that is found in all 3 forms of life. So acetylation is basically mean you have acetyltransferase that transfers the acetyl group from acetyl-CoA to protein. It could be at N-terminus, could be alpha position of N-terminus amino acid, or could be on epsilon position of lysine residue. So these 2 kind of but N-terminus acetylation is reversible, but irreversible, and epsilon acetylation is reversible. So it involves in the regulation of many cellular functions like transcription, translation, and gene regulation, and protein interactions. So pylori infections, half of the world's population is affected with pylori, but though the disease is not manifested, but you have pylori in your stomach. So HP0935, the putative acetyltransferase is an essential gene for pylori, without which it cannot survive. So we are interested to characterize it and trying to drug it if possible. Thank you.
How big are the cells?
This is 18K. You have variety of sizes.
Good afternoon, everybody. My name is Christian Ribiela. I'm working at the lab of Dr. Neil London of the Weizmann Institute. The main focus of our research group is to find small molecules that covalently bind to protein targets. And in my case, I'm working on the prolyl isomerase PIN1. Why do we want to target PIN1? Because it's a key element in cell signaling and therefore a potential drug target. So it's abnormally upregulated or overactivated in about 60 tumor types. And it has a very good targetability. The active site has cysteine 113 that can be attacked by covalent inhibitors, electrophiles. And there are some existing inhibitors, but they have a lot of problems with solubility, permeability, cell permeability of targets, and so on. And so this is why we're looking for research tools that could be used to investigate this very interesting enzyme. And what we achieved so far is we identified a hit using an MS covalent library screen, and we were able to optimize this hit by using computational chemistry. So we had docking studies, and we evaluated our candidate probes in biochemical assays. And right now what is missing is X-ray, just structural information to first validate our docking studies, which our lab is constantly working on, and also to further optimize our probe to finally be able to make ABPP activity-based protein profiling. And so our goal is to find a PIN1 probe, and it has to be selective, active in the nanomolar range, and so cell permeable. Thank you for your attention.
So you look at, okay, this Engelbert is not here.
He's not here?
Apparently not.
Come tomorrow.
Tomorrow. Thank you for filling in. Great.
Hi, good afternoon. Myself Vijay. I am a postdoc at Dan Taufik's lab in Weizmann Institute of Science. So my work basically involves evolution of enzyme regulation. So what do I mean?
The microphone.
So what do I mean? So if you take any enzyme family, it will have different members. So some may be unregulated enzyme, very simple enzyme that has just converts the substrate to the product. And there may be other members with allosteric binding sites. There may be other more members which has heterooligomerization, and also different oligomeric state that confers cooperativity in enzyme activity and other regulatory features. So my basic question is to answer how these regulatory properties evolve. So if you start from an unregulated enzyme, and what are the mechanisms, like what are the physiological constraints, what is the evolutionary mechanism, and what is the structural basis of these regulatory properties. So these are the basic questions that I am addressing. So how I am going to do it? So the model system that I am going to work on is glutamate dehydrogenase from Bacillus subtilis. So there are two paralogs in this organism. One is called ROCG and one is called GODB. So ROCG is, both the enzymes are functional as hexamers, but the key difference is the ROCG is a constitutive hexamer, whereas GODB becomes a hexamer only if you add glutamate. So it's a glutamate induced assembly of the hexamer. So we have found growth conditions in which the only GODB expression gives a growth advantage, not the ROCG. So basically I will use this model system and I will start with a ROCG, a gene expressing ROCG. I'll make libraries of this enzyme and then use this selection system to find out whether I can convert this ROCG, which is a constitutive enzyme, into a regulated enzyme. So, all the evolutionary intermediates that I get in the selection, I'll be subjecting it to kinetic analysis, structural analysis, as well as thermostability analysis. So yeah, that's the study. Thank you.
Hello, my name is Pandora Karakousi. I am about to complete my master's in microbial biotechnology, and I plan to continue for a PhD in structural biology. Firstly, I would like the organizing committee to give me the opportunity to participate in this workshop. Since last November, I work in Dr. Evangelias Christina group in the National Hellenic Research Foundation, and in the frame of collaboration with Professor Hadjinikolaou from the University of Athens. My project involves structural studies of 6-phosphobeta glycosidase of GH1 family, applying the method of X-ray protein crystallography. The role of our enzyme is to hydrolyze phosphorylated oligoglycans for further utilization by aerobic soil bacteria. We show that it exists a broad specificity of 6-phosphoexosis, and it will be happens in high temperatures. Cloning expression and purification resulted in a high-resolution enzyme, in the high-purity enzyme, of a molecular weight of 55 kilodalton, suitable for structural studies. Enzymatic and activity in the enzymatic thermostability studies shown that our enzyme is optimally active at 65 degrees Celsius and retains more than 80% of its maximum activity at 70 degrees. Crystallization trials, crystallization experiments are carried out in our structural laboratory at NHRF. Here we can see, Here we can see the first crystallization trials for commercial screens after rational selection, and further optimization resulted in rhombohedral shaped crystals. All crystals are tested in our house X-ray facility, robotic facility, and we confirmed that they are protein crystals, but the resolution was too low for preliminary characterization. So now we are working to improving the size of the crystals, and we aim to collect high-resolution data from synchrotron radiation sources in one of the European facilities, such as CSRF or Synchrotron. Yes, thank you.
Hello to everybody. Myself Shobhan Kuila from New Delhi ICGEB, and as I joined my recent lab, I got interested in what I'm eating. So every time I eat, I take a count of how much vitamin C it contains. So it is because our daily lifestyles has become induce several stress in our body. So every stress leads to an oxidative stress in each cell. So to overcome this oxidative stress, we need lots of antioxidants. So vitamin C is considered as a major antioxidant because it's small and soluble and abundantly present in all the vegetables, fruits, and freshly meat and raw meats. So as a human, we have lost the ability to vitamin C. So we solely depend on the dietary source and recycling. Yes, so our protein shown to have recycled vitamin C in our body. Don't go by the name, it's human chloride intracellular ion channel because it is found as, initially it was found as an ion channel, but no structure available yet. But it is produced as soluble form, which is now recently shown that it recycles vitamin C. Also we found that this HS-CLIC1 is differentially overexpressed in several cancers. and we know cancer cells is under high oxidative stress because of huge demand in cell cycle replication. So we are exploring this its structure. Structure is already available with us, the soluble form. We know the enzyme active site, so we try to specifically block that enzyme through its active site, so that in a hope we can target cancer. At least we will know what's the role of vitamin C recycling in cells. So thank you.
I have a question. You have a photo saying bound compound in crystal complex in your...
Sorry?
Go back a slide.
Go back
yeah. So
this is
your
experimental result? Or?
Yes. So how we are targeting this? We know that the active sites of the enzyme. We are specifically looking for compounds which specifically block. It contains a cysteine in its active site. So we are specifically trying to block this with a covalent modification and through molecular docking. And we got some compounds initially, and we tried co-crystallization. So one of the structure is there. We got only one with this. This is not binding at this position. It's binding somewhere else. Maybe allostatic sites.
It's not in the active site.
It's not here. It's some binding somewhere else.
I see. Okay.
So now we are trying with the enzyme assays and cell line testing, so that maybe we can come up with something. And this structure will help us in making more specific and more less side effects. So thank you.
Hello everybody, I'm a postdoc in Indian Institute of Technology, Delhi, and my mentor is Dr. Ashok Kumar Patel. So basically I am working on pyruvate kinase 2, which alters the glucose metabolism in cancer cells. So how it alters the glucose metabolism, the PKM2 interact with the hypoxia-inducible factor, this HIF1 alpha. So like one enzyme is PHD3, pyrrole hydroxylase 3. It alters the pyruvate kinase 2 structure, and it activates the PKM2 to interact with the HIF1 alpha. And HIF1 alpha induce some transcription factors like glucose transporter and lactase dehydrogenase. So we are targeting the when it PHD interact with the PKM2, how that structure will change. So we are crystallizing this complex. So we have purified this PKM2 PHD complex from GFC. Then we confirm it on Western blot. Then we also check on native. So here's the PHD3, then this PKM2, and then this is the complex. And these bands also from SDS we have confirmed by MALDI. So these complex we have tried for crystallization, and we got some hints. So we have got some crystals. So these crystal I want to try in this workshop. Thank you.
Did you send the crystals?
Yes.
Great. So this is the first time you're testing them?
Yes, we are testing first time. This is the complex. This is the complex crystals.
You didn't check on any home source yet?
No, not yet. So we have run the gel actually in our SDS. We have checked by silver staining. We have taken while freezing. Few we have one, and I have taken on the check on staining this silver stain.
And you confirm that you have the complex now?
Yes.
Did you use the crystal to run it in the native gel?
No, not native. We have confirmed on the silver stain.
Silver stain.
Is it enough for one crystal to do a gel?
So it's on SDS. We can't we can't see. So but silver staining we can see the. Thank you.
Thank you.
So good afternoon everyone. I'm Devanshu from India. So I'm also working on a small heat shock proteins from protozoan.
So as Sripanna discussed that small heat shock protein are ATP independent molecular chaperones. So these small heat shock proteins are low molecular weight ATP independent molecular chaperones that has a property of forming higher oligomer that ranges from dimer to up to 48 mer they can form, and this small heat shock protein has 3 domains: N-terminal domain and N-terminal domain, C alpha crystalline domain, and the C-terminal domain. So alpha crystalline domain, that is the most structured part of the protein, which is flanked by N and C-terminal unstructured region, and and it has the property of forming a dimer, which is the basic functional unit of this protein. And there are 2 types of dimerization found in small heat shock protein. The one which found in this non-meta zone, in which that the loop between the beta 5 and the beta 7 strand interact with the other other monomer unit, while the other dimerization found in meta zones, in which there is an extended beta 6 beta 7 strand which interact with the beta 6 beta 7 strand of the other subunit. So what happens, and the function of this protein is to prevent aggregation during the stress condition. Like when there is a stress, the other protein start aggregating. So to prevent it from aggregation, this small heat shock protein goes to the unfolded protein and binds there. When the normal condition comes, it may this protein accessible to the ATP dependent molecular chaperones and helps in folding back to their native state. So this small heat shock protein has a role in diseases like cataract. So currently I'm working on parasitic protozoans like Entamoeba histolytica and Plasmodium falciparum. Other than this, I'm also working on Oryza sativa. In Entamoeba and Oryza sativa shows chaperonic activity up to 53 degrees Celsius and 48 degrees Celsius respectively. By the I check this chaperonic activity by NDE1 Thank you.
Those are your structures there?
No, sir. This is the PDB.
Okay.
Hi, my name is Michal Isnyansky. I am MD PhD student. I am from Tel Aviv University, and this study is conducted under the supervision of Dr. Yoni Chaitin and Dr. Moshe Giladi. Okay, so brief background. Osteoporosis is a skeletal disorder in which the bone density is reduced, and this situation leads to bone fractures. In order to prevent these fractures from occurring, it is widely common to use pharmacological agents, which are called bisphosphonates. Known yet very strange side effect of bisphosphonates is the appearance of atypical fractures. And by saying atypical fractures, I don't mean osteoporotic fractures. I mean fractures which are caused with minimal trauma, if any. They appear in different places along the bone and in different severity. The molecular mechanism by which bisphosphonates prevent osteoporotic fractures is by inhibition of enzyme from the mevalonate pathway, which is called FPPS. But recent study showed association between mutation in other enzyme in the mevalonate pathway, in GGPS, which I study, and the appearance of atypical fractures under this treatment. We wanted to understand the molecular mechanism by which this mutation in this enzyme leads to atypical fractures under this treatment. And to do so, we started with the structural characterization of the enzyme. Actually, the wild-type structure was previously solved by this group, and we crystallized and solved the crystal structure of the mutant enzyme. This is the apoform, and this is the bisphosphonate-bound form. You can see here the binding site. This is actually hexamer, and this is the binding site of the apo, and here is the binding site with in the bisphosphonate-bound form. You can see that the mutated tyrosine is not participating at the binding of the bisphosphonate. And after the structural characterization, I performed a kinetic assay in order to characterize the activity of both enzymes. you can see that the catalytic activity of the mutant enzyme is lower in comparison to the wild type. And after doing so, I performed thermal shift assay, which is the last experiment which I showed here, in order to assess the KD of both enzymes toward this drug. Okay, thank you.
These are all your structures?
Yes.
Where did you collect the data?
Yes, sir.
Shalom and good afternoon to everyone present here. I'm Sudarshan Murthy and I'm a mid-phase PhD student in Faculty of Biochemistry and Molecular Medicine, University of Oulu, and my supervisor is Larry Letthew. Today's topic, basically I'll be talking about the development of small molecule inhibitors for MATIDs. Our group is basically a structure-based drug discovery group, and then we are focused on developing small molecule inhibitors for the entire family of this ARTDs, human ARTDs. So the obvious question would be, what are ARTDs? So ARTDs are enzymes which catalyze a reversible post-translational modification, that is ADP ribosylation. So by doing that, the ADP, they change the properties of the target protein, may it be the enzyme activity or maybe it be changing the binding partners and stuff like that. So in the reaction, NAD is cleaved to nicotinamide and ADP ribose is added to the target protein. Today I'll be just discussing about the human ARTDs, and in this superfamily there are 17 enzymes. They are differentiated based on different categories, and the major category would be based on activity, and if you categorize them based on activity, they're classified into 3 subgroups, that is PARTDs, which are ARTD 1 to 6, and MARTDs, which are ARTD 7 to 17, and the inactive ARTD 13. And my project solely comprises on the MARTDs. So MARTDs are subclass of this superfamily where they transfer only one ADP ribose to the target protein and in turn changes the property of them. MARTDs were recently discovered, say about a decade ago, so most of its role and functions are still very elusive. So, and also another problem is that there are no specific tools to verify their roles. So with all this hindrances in the way, there's very little known about the MARTDs, and with this we aim to develop selective inhibitors for specific members of MARTDs. With this, we think that they can act as an important tool to evaluate cellular function and also help us elucidate the ADP ribosylation signaling. And also this can act as a dual function, like they can be a starting point for drug discoveries, as recently MARTDs have been shown to be involved in cancer biology, and so they're a hot topic. And in this MARTDs subclasses, the focus is on ARTD10. ARTD10 was first identified as a binding partner for oncogene CMYC, and then it also interacts with PCNA and actively participates in S-phase DNA damage repair. They also, is it time? Oh shit, okay. Off the record, please. Okay. So with these important targets, we started screening libraries, and going back in history, one of my predecessors who were working on this project, ARTD10, he screened the library from the National Cancer Institute, and he found a hit molecule that was OUL, that we named OUL 35, which has 2 benzamide kind of moiety linked by an oxygen linker. And this compound is the first and the most potent at least as of now, it's the most potent ART-D10 inhibitor. So with this as a starting point, we went ahead to do structure-activity relationship of the same compound, and we used a traditional approach where we kind of modified the parent compound in different parts and tried to find better and more promising compounds. In this search, we ended up with a compound which we named compound 32, which is much better than OUL35 with a potency of 200 nanomolar, and also I've shown here as a thermal shift where we can see a good shift in the oh shit. Okay. yeah. So I've also shown here the shift here, 5-degree thermal shift. So promising compound.
Yeah. Hi, I'm Naveen Narayanan from Dr. Deepak Nayar's lab in Regional Center for Biotechnology. I work on a small protein called as a MOM, which is actually an abbreviation for modification of mu. So mu is a temperate phage that infects E. coli, and at the late lytic phase, this MOM protein gets expressed and it modifies the genome of the phage, so that it's protected against the restriction modification enzymes from the bacteria. And what we could see that it transfers a group called acetamido group, and till now we don't have any structures of an acetamido transferase or anything that the mechanism of acetamido transferases mechanism. So it transfers the acetamido group at the N6 position and N6 position of an adenine, and renders a genomic DNA resistant to the cleavage by restriction endonucleases. So we aim to understand the mechanism by which it acts. So I got the structure of this particular protein, I got the crystal, it diffracted to around 1.5 angstrom, and before that we couldn't solve it, but now we got the selenomethionine crystals and we could solve it. But the major question is that what is a cofactor that is acting? So we don't know, whatever structure we have doesn't have any cofactors. So I started by soaking with coenzyme A. We also have another way by which we could take the cell lysate and then concentrate it and then add it and soak with those crystals, soak it with the crystals and see what's happening. So the main objective is to understand the mechanism by which it acts. And we're also planning to get the structure with the DNA, so that could solve a lot of the problems. So that's it. Thank you.
Did you ever try to purify the protein and then do mass spec to see the cofactor that way?
No, there is no cofactor. I think it's not getting bound with that thing. Maybe it requires a DNA bound structures to get it with the cofactor. So that's why we wanted to shift into DNA bound crystallization also. But we are trying with acetyl CoA and CoA and all those things, which is supposed to be theoretically the best possible cofactor that is possible.
Okay. Good afternoon, everyone. My name is Sivaji. I am working with Professor M. Vijayan at Molecular Biophysics Unit, India. So our lab extensively, from the past 4 decades, we extensively worked on lectins from plants and their specificity towards sugar. So very recently, a decade ago, we shifted our focus to microbial lectins such as mycobacterial species, and then recently I started working on archaeal lectins. So to briefly introduce the lectins, so these are the class of carbohydrate binding proteins which specifically recognize the sugar moieties and then thereby perform their actions. So lectins in animals, plants and bacteria have widely reported, but there were no existing reports in the literature about archaeal lectins. So very recently we had done a bioinformatics study by applying sequence comparison and then fold recognition tools and identified reputative lectins in archaeal kingdom, and I started working on doing experimental work on this, some of them. So subsequently I was able to purify and then crystallize and characterize the lectin biochemically, and then so I got the crystals. So they crystallized in tetragonal space group, but still I'm trying to solve the structure using molecular replacement, and then I tried using cell-mate crystals also. Still the structure solving is in progress. So by looking at the preliminary data we have, and we compared the sequence with a known three-dimensional database, the sequence identity very less, is coming close to 20-22%. So we think that in a fold wise, so these lectins, archaeal lectins, are a little bit different than what existing in plants and animals. So, and then the most important point we are trying to address here is that by looking at these folds and the structure of the lectin, so we wanted to see how these lectins are evolved through the evolution from archaea to higher eukaryotes. Thank you.
So, hello everyone, can you hear me? My name is Johan Pääkkönen. I come from the University of Eastern Finland. My project is the Caulobacter crescentus xylonolactonase, which is an enzyme that catalyzes the hydrolysis of xylonolactone to xylonic acid. This reaction and this enzyme belongs to the metabolic pathway of xylose sugar to xylonic acid, and ultimately to other interesting products such as 1,2,4 butane triol or ethylene glycol, depending on the enzymes. This has a lot of industrial potential. So I've been able to crystallize it and optimize the crystallization conditions to obtain good crystals. And the crystals have been measured at both DLS and ESRF synchrotrons. Best data set being of resolution 2.8 angstroms, so not very great, but it allows us to solve the structure preliminarily. So using molecular replacement with a homologous gluconolactonase, I've been able to solve the structure, and the tertiary structure is a beta propeller in which there's a metal binding site and the active site here in the tunnel. But the resolution yet is, I'm not saying bad, but bad enough that we cannot see the metal binding site well, meaning that the metal and the side chains are not visible in electron density. And also this is not a complex structure. So here on this course I will try to, or I hope to be able to get a better result with new crystals.
Thank you. Crystals?
Yes. Yes, thank you. Any questions?
What is the metal? What is the metal?
We are not sure about that. We are trying to find out.
In the homology protein that you used, what metal was there?
I think it was calcium.
Hey, good afternoon everybody. I'm Jakub Táček from a Laboratory of Structural Biology at the Institute of Biotechnology in Prague. And my project here is the comparison of sulfamides and phosphinates as bimetallic peptidase inhibitors of glutamate carboxypeptidase 2. Glutamate carboxypeptidase 2, GCP2, is a protein. It's a membrane and zinc-dependent neuropeptidase, which can be found on the surface of astrocytes in the nervous system, and it cleaves one of the most abundant neurotransmitter, and it releases glutamate. Released glutamate, or in some neurological disorders, is a reason why is a cause of these disorders when there is excess of this glutamate. So apparently we aim at inhibition of GCP2 because it in such neurological disorders, in such conditions, it might lead to the lowering amount of free glutamate, and so it has medicinal potential, which was already proven in some mouse models. So we aim at the inhibitors. Basically, inhibitors are based on its natural substrate, which is basically the peptide. So this is the scheme of the general inhibitor. There's one part which is the glutamate. Because this is a GCP2, it is highly specific for the glutamate part in this position, so there's not much we can do about it in inhibitor design. Then there's another part which is which comes from a peptidic bond of the neurotransmitter, but there has to be some peptidic surrogate which is non-cleavable and which will bind to the two catalytic zincs in the active site. And there's this other part which is in the entrance tunnel to the enzyme, however, but I will not talk much about it. There can be made many changes. So what we aim at right now are these zinc binding groups. Here we can see some series of several of them which are usually used for this purpose, and we have already tried some of these. We have also some crystal structures of some complexes. However, right now we are focused at the phosphinate and sulfamide. These are the two most interesting zinc binding groups for us, and there is a difference between them. Phosphinase generally has a better potency. However, potency is not the only thing you care about in case of the inhibitor, because in this case, as I said, GCP2 is in a nervous system. So if you want to administer the inhibitor, it has to cross the blood-brain barrier. As I said, there's a glutamate here which is highly polar. So we are looking for some, and there's not much we can do about this. So we are looking for some these zinc binding groups which will be less polar and increase the lipophilicity of this inhibitor to be able to cross blood-brain barrier. So phosphinates are pretty good compared to sulfamides. Their potency is a bit worse. However, they have much better pharmacological properties. So, however, there are no structures yet, and we were surprised a few times about the binding mode of inhibitors. So right now we aim at the solving crystal structure of these complexes of both these inhibitors, and to use this for some future structure to the relation studies design of new inhibitors. Thank you.
Hi, good afternoon everybody. So I'm Ishan. I work under Professor Prasenjit Bhaumik, and I work at the Indian Institute of Technology, and this is the department, and we are quite fortunate enough to have all the facilities to do the work there and collect data. Few data sets were collected at Indian Synchrotron and few at ESRF as well. So I work on a protease which is plasmapsin, which is from Plasmodium falciparum. It has also been proved to be a good drug target, and my work is to understand the structural insights into the activation mechanism of the protease, and also to look at how the peptide binds in the active site of the aspartic proteases. So since this is a aspartic protease and this is a protease, so it is synthesized as a zymogen initially, and so it is not active. So with Sachs experiment we could prove that this forms a dimer, and even in the crystal packing we see that all the structures that have been solved previously and that we have solved in our lab, we see that it forms a dimer. And when we superpose all the dimer, we see that there is actually a movement in one of the dimer, and to understand what was this movement related to, we look at the structure and we see that this particular region which is at the dimer interface, we see that this particular interaction which is there, if it gets disrupted, we see a huge change in movement of these dimers. And based on this results and few other results that I'm not shown here, we could try to propose a mechanism, and this is the mechanism that we propose, how the zymogen converts to the mature form. Here what we see is, we see that initially they are in the dimeric state, and under acidic condition they do get activated. So when you put them in acidic condition, this kind of interactions break apart, and then it loosens the dimer interface. The area in the dimer interface is reduced, and this converts to the monomer. So once it converts to the monomer, the mature enzyme which is there can actually come here and cleave the zymogen and convert to the mature form. So this is not completely foolproof. We are trying to figure out some of the few experiments that are remaining to prove the mechanism. So this is one part of the study. The other part of the study is to understand how the peptide binds in the active site of aspartic proteases. Since aspartic proteases are unique because they have two aspartates, and we don't know which one is acidic and which one is neutral. So we wanted to probe using peptidomimetic inhibitors. So we have a set of peptidomimetic inhibitors which are modified subtly at certain positions. So we solved for 3 crystal structures here with this inhibitors, and interestingly in this one we see that we change this very similar to the peptide substrate for plasmapsin. We see that the inhibitor flips, so it is actually oriented opposite to the binding of the previous inhibitors. And based on all these 3 structures, we also see that it has been believed in the aspartic proteases that they are actually planar, but we have seen that they can be non-planar, and we have seen other structures as well where they have not commented on the planarity of these aspartates, because this planarity could actually be helpful in determining which aspartate is more important during catalysis. And based on these results, we have tried to propose few things which are quite unique to aspartic proteases, which was not known, and I hope to discuss few of this in the poster session tomorrow. And any questions I would happy to answer. Yeah, thank you very much.
So hi everyone, my name is Efrat. I'm here from the Weizmann Institute. I'm doing my PhD in the lab of Dr. Neil London. And in this project, we're trying to rescue the activity of a mutated version of p53 protein. So a lot of you probably know that p53 is a very important tumor suppressor. Upon some kind of stress, like UV radiation or toxins or something like that, this protein will be activated, and in turn it will go and bind specific parts on the DNA and activate different pathways, DNA recovery pathways or apoptosis and things like that. So the specific mutated that I'm researching is not able to bind DNA. And if you look at the structures, you can see that its structure is very similar to the wild-type structure, but it still is not able to bind DNA, and this is because the mutation is in the interface between p53 and the DNA. So in our goal, what we're aiming to do is we are developing a compound that will be able to bind specifically to this mutated protein, and by doing so, we'll reactivate it and mimic the wild-type protein. So Chris from my group, as previously a bit mentioned it, we're using covalent compound to do that. We're screening using LCMS, and I hope that by the structures that I sent for this workshop, we will be able to see some more insights upon the binding of the compounds that we have, and further improve our recognition element and our activation of the compound. Thank you very much.
From the structure, that was probably modeling. From the modeling, can you see why it doesn't bind to the DNA?
So
that was by modeling.
Yeah. So here in green is the wild type protein. So it interacts with the DNA, and with the mutation, it cannot do these interactions.
Is the mutation at that residue?
Yeah.
Okay.
גם לא נמצא. ניר לא נמצא גם. Hello everyone, my name is Anat, and I'm working at the Macromolecular Crystallography Research Center at Ben Gurion University, which is located in Be'er Sheva, which is down south Israel. My talk will be slightly different because the MCRC is the service center, and therefore I don't have a project of my own. but I do I do involve in the structural parts of many projects throughout the university. So what I show you here is all the facilities we have in our center. We start with the is there a pointer? Yeah. We start with the experiment design and design the plates. We have a special software for that, which we can design whatever we like. And after the experiment is designed, we take it to it's a drop setter robot, which is similar to the Mosquito, which I assume most of you are familiar with. It's a robot made by Formulatrix. All our robotics actually are of Formulatrix. And after the plate is set, it goes to the imager, which can accommodate 1,000 plates. And the advantage of it, it has a regular camera and a UV camera. So if you have protein crystals, it can be an initial indication that this is indeed protein and not salt crystals. And we also have a website in which you can see and follow after all the crystallization plates, which is very convenient instead of looking at them under the microscope. And if we saw like an initial heat that was shine in the UV camera, and we saw it in the website, we take it to another robot that we have, which is called the formulator, and it do an optimization of the crystallization screens, which is also very convenient. It can do both 96 well plates and 24 ones. And if we have proper crystals that are big enough, we also have two instruments like this, which are like X-ray for the lab systems. Although we did collect many data to resolution around 2 angstroms. And we also go to Synchrotrons with all the projects we have in the center. That's it. Thank you.
Good afternoon. My name is Joanna Śmietańska, and today I'm going to tell you something about ANS complex of hype 1. It's a Synchrotrons word PR10 protein with non-crystallographic symmetry and tetrahedral twinning. Currently, the main object of my research are macromolecular modulated crystal structure, such as hype 1. It's a pathogenesis related class 10, PR10 plant proteins obtained from medicinal herb Synchrotrons word. According to current results, the hype 1 protein is involved in phytohormone binding. The main PR10 fault of hype 1 protein consists of a seven-stranded antiparallel beta sheets wrapped around the C-terminal helix alpha 3. Our earlier studies shown that hype 1 protein can co-crystallize with ANS, highly fluorescent dye ANS, can obtain produced crystals with tetrahedral twinning of C2 unit cell with an apparent 9-fold translational non-crystallographic symmetry along the main C-axis and containing 36 unique protein molecules in the expanded supercell. Although the structural modulation is widespread and well understood in small molecule crystallography, in macromolecular crystallography this phenomenon is practically unheard of. We can distinguish 8 ANS bonding sites in our crystals. Sites 1 to 3 are located inside the protein cavity in the internal chambers and the and the deep pocket surface, respectively, whereas the other 5 sites are involved in the surface-bound ANS molecules interacting with nearby residues. There is a hypothesis that size of the unit cell and structure modulation can be dependent on the amount of ANS ligands. So the next step of our project will be to determine the impact of amount of INS molecules of the structure modulation, and our research are currently in progress. Thank you for your attention.
Good afternoon, everyone. I'm Güler, and my supervisor is Burcu Kaplan Türkes. I'm from Turkey. First, I want to talk about myself a little bit. In my country, there is a few people just work on crystallization. And when I doing my undergraduate lesson, I realized the importance of know the structure of the biological material. And then I found my supervisor, Burcu, and I said I want to work with you, and she was so happy. Then I started to work on crystallization, but in my country there was no source like the chemicals or materials. Then I was trained on sample preparation and characterization laboratory at EMBL Hamburg, and I just learned something like how can do a crystallization, how can prepare my sample. Then I go return my university, and I start my project. My project was on production, purification, and crystallization on Ximomonas mobilis levansucrases. Levansucrases is a bacterial enzymes. The atomic structure of several microorganisms were solved, but difference between them and my enzyme is that some of them just can produce levan, and some of them just can produce fructoolisaccharides, the different product. But my enzyme, Ximomonas mobilis levansucrase, can produce both. We don't know the reason yet. By solving the atomic structure of it, we can understand the reason of difference at molecular level, and maybe we can control and change it, because this product levan and fructoolisaccharides is important on food industry. I'm a food engineer, by the way. It's really important things on food industry. Then I managed to produce the wild type enzyme. Then I purified it. You can see my purification process. It was really hard to purify because it's wild type. I just tried so many conditions. I changed everything by time. Then after purification, I made some experiment to prove this dual activity. It can produce levan and phos. Is it true or not for my enzyme? Then I prove it. It have dual activity. Then I used the anion exchange column. I was purified my enzyme. Then after that, I set up some plates, and I couldn't see the crystals. Then I go further away. I use size exclusion chromatography, and I was managed to get them to take monomer form of my enzyme. It was really important for me, and with this monomer form of enzyme, I set up another plates. Now I'm waiting for the results. So far I don't have crystals. Then I'm waiting to results, and also, by the way, I realized something. The zinc prevent the proteolysis in the presence of trypsin. Then I start with the zinc as a starter conditions. And also I have a poster and like the small form of it. For further information or details or see my results, you can take it. Thank you so much.
Hello everyone, I'm Shruti from University of Olu. I work under Rick Viranga. So I work on project of studies of protein crystallographic of substrate specificity and substrate channeling of rad peroxisomal multifunctional enzyme type 1. So this is the crystal structure of the wild type, which is bound with NAD and CoA. So the rad peroxisomal multifunctional type 1 enzyme is a multi-domain protein with 5 domains differentiated as A, B, C, D, E. So it catalyzes the second and third step of the beta oxidation of the fatty acids. So it is a monomeric protein of 80 kilodalton and is of 722 residues. So there are 2 full-length constructs which we have made, that is A, B, C, D, E and the B, C, D, E. So I will tell you what are the 2 constructs. So the protein has 2 enzyme activities. The first enzyme active site, the N-terminal domain, is called the crotonase fold, which catalyzes the hydration of the enoyl CoAs. The second, or the C-terminal domain, is called the HAD fold, which catalyzes the dehydrogenation of the 3-hydroxy CoAs. So the catalysis is, So the catalysis is where the 2-enoyl CoA is converted into 3-hydroxy in the crotonase fold, and then the 3-hydroxy is converted into 3-keto in the dehydrogenase fold. So in this protein, I have also carried out studies of substrate channeling. Now substrate channeling is reported for this protein, and I've been carrying some assays for that. And we have also trying to find out, is there a flexibility important for this mechanism, and I'm also trying to find out if there is some substrate specificity by fragment screening and other studies. Thank you.
Good afternoon, everyone. I'm Ramita Sulu from University of Olu, and today I'm here to give short introduction on my project. I'm working in characterization of catalytic and structural properties of human collagen prolyl 4-hydroxylase. It shortly calls also CP4H2. CP4H is a central enzyme in the biosynthesis of a collagen, which catalyzes the hydroxylation of proline in XPG sequences of procollagens. This process is necessary for the conformational stability of a collagen. And regarding the molecular structure of this protein, it is a heterotetrameric structure of 2 alpha subunit and 2 beta subunit. The beta subunit is identical to protein disulfide isomerase, also called PDI, and the alpha subunit is catalytic subunit, which is further divided into 3 domains. And there are 3 forms of CP4S, isoform 1, 2, and 3. And till today, the crystal structure for this N domain and PSP domain, and also the full-length beta subunit has been solved. But there is no crystal structure for this cat domain and as a whole full length of a CP4S. However, we have currently solved low resolution structure for the full length CP4S with the help of SACS, and this is the current working model for CP4S full length. And so now our ultimate target is to get a high resolution three-dimensional structure for full length, as well as to understand the interaction between the cat domain and this beta subunit. So for that we have generated different construct of CP4S, and among which CP4S to delta 260 is a variant which contains the catalytic domain of alpha subunit and a full length beta subunit. And we are able to get crystal for this construct, and which diffracted at 4.5 Armstrong. And in order to check if this crystal contains our both subunit or not, we have ran the crystal in a STS, and that confirmed that it contains both subunit. And I would like to focus at the end that this is the first crystal with alpha and beta subunit together, and from this crystal structure we can get understand not only the catalytic mechanism of catalysis of this enzyme, but also it can be used to develop the drug for treating many collagenous diseases, including fibrosis, as well as the many metastatic cancer such as prostate cancer and breast cancer. So if you want to know further, you can check on my posters. Sorry. Sorry. Okay. Thank you.
So, hey, I'm Sibel. I studied biochemistry in Germany, and then I moved to Sweden to study biomedicine, but it's medical structural biology. So I work with superantigens, and so it was found out that they bind to a cytokine receptor, which was called GP130. And I investigate the binding with surface plasmon resonance and try to crystallize the complex. So, what is a superantigen? I think you know what a normal antigen is. An antigen binds to MHC plus 2. That one is presenting the MHC plus the antigen to the T cell receptor. So, to the T cells, to the T cell receptor. And then you get a normal immune response, cytokine release, and so on. And a superantigen is binding differently to these both proteins. It binds simultaneously to MHC class 2 and TCR, and you get a massive immune response. But that was all investigated. There are a lot of structures of superantigens and T-cell receptors in MHC class 2. So now it was also found out with surface plasmon resonance that they bind to GP130. And we purify a truncated version of GP130 because there was most of the cytokines, there are 10 different cytokines so far known to bind to GP130. And we see it also with surface plasmon resonance that it's binding. And the interesting thing is that it's only binding to the human GP130. So we tested mouse and rat and it's not binding there. And they have a sequence identity of 74%. So we're also thinking about computational modeling where the sequence differences could help us to see where it could bind, because the binding is also zinc dependent, which is also a limiting factor. And so GP130 is a surface signaling receptor, and it binds cytokines of the interleukin 6 family in 10 different ones with slightly different structures. So, but we don't know what the binding is really doing. We know that the binding is between the domains D1 to D3, but we don't know if it's only blocking the normal GP130 pathway or activating one of the signal pathways. And yeah, my task is to determine the X-ray structure and to do more surface plasmon resonance experiments to see maybe if they're competing or binding on one side, which is already known. Yeah, and so far, so this is my first year, I am able to produce both proteins. GP130, like not a lot at all, so but I'm still trying to optimize it with different constructs. But the superantigen is fine now, and I have an initial crystal hits, but they are not diffracting, so I thought it's a bit of a waste to ship them to Israel. So I didn't bring any crystals, and I tried to get my constructs working so I can get better, more protein, and yeah, get better crystals. That's it.
Hello everybody, I'm Bo Wang, I come from Peking University. I feel some nervous today because this is the first time I take a presentation with English. So please forgive me if I make some mistakes. My university is the oldest university in China. This year, my university is greeting its 120 founded anniversary. So many activities and ceremonies are setting up on the campus. My university has a very beautiful campus like the Weizmann. This picture was taken by myself about 1 month ago, okay. And I will share something different with others today. I will share some experience about the EM on the Synchrotrons workshop. HIS1 is a very important protein in plants. I want to solve the structure of the HIS1 at first, but I can't obtain this protein. I can't obtain the purified HIS1 because it is always bound to the GROYAL. As we all know, GROYAL is a very important chaperone in many kinds of bacteria. When I express the HIS1 in the E. coli, the HIS1 always bound to the GROYAL, but we can't find the GROYAS. So we infer that there may be some new mechanism about how the GROYAL works in the bacteria. We have to solve these problems use CRO-EM because it is very powerful tools to solve the structure of the big molecules. So a lot of negative sting tests were conducted, and fortunately we found that there may be some conformational change after HIS1 bound to GROYAL. We can see that that picture is the circular particles were changed into the cubic particles, and we operated particle clarification and 2D average using about 60,000 particles, and we can see more clear particles on this picture. And next, we choose some good particles to set up a 3D initial model. This picture is showing the initial model, and this figure is wrong. It's 20 angstrom, and compared to the model from the PDB, unknown density was founded. Yes, it's here inside the cavity of the GROYAL. So up to now, we are collecting some data sets under the CRO-EM. So the data processing and analyzing will be conducted in the future. Thank you, everyone.
One more talk. We have one more talk without slides.
Is he around?
Yeah. I asked him, he didn't get his slides in time, but he will give a talk anyway. Short talk.
He did for competition.
Okay. Hi, good afternoon. My name is Fadi Jbara. I'm a PhD student in Professor Abid Azim and Professor Joel Hirsch labs. Following the synthesis of proteins as linear chains of amino acids to become fully functional, they need to fall to 3D unique structures. Fundamental insights into the folding process was provided by Vincent. I think it was like very fast. Fundamental insights into the folding process was provided by Vincent's pioneering experiments, which showed that small protein can fold spontaneously upon removal from the naturant. However, later it became clear that small proteins and that test fold conditions work only for small proteins and in conditions far from those encountered in the cells. Large proteins and proteins with complex fold topologies like 10-fold barrel tend to populate kinetically trapped intermediates, which expose hydrophobic amino acids into the solvent and thus are prone to form aggregates in a concentration-dependent manner. Indeed, cells have evolved a family of proteins referred to as molecular chaperones that assist protein folding primarily by preventing inappropriate interactions between non-native polypeptides. HB60 is a ubiquitous molecular chaperone found in all kingdoms and are distinguished into two groups. In our lab, we investigate the mechanism of action of the human mitochondrial HB60 by crystallography and cryo-EM. We solved the structures of 3 different intermediates in the cycle of HB60. The crystal structure of the football with ADP and beryllium fluoride, the cryo-EM of the football with ADP, and the cryo-EM of the half football with ADP. Obviously, in the remaining of my short talk, I will concentrate on the crystal structure of the football with ADP and beryllium fluoride. The main obstacle in obtaining highly diffracting crystals with HB60 is the labile nature of HB60 as it exists in equilibrium between heptamers and monomers. Thereafter, we utilized a much stable version of HB60, which exists solely as a heptamer. We set out hundreds of crystallization trials with the stable version of HB60 and the helper of the HB60, the HB10, with ATP and beryllium fluoride. The purpose of beryllium fluoride is to stack the complex in the prehydrolysis state. We got, in one of the conditions, tiny crystals. After rounds of optimization, we extended the resolution to 6 angstrom. With the full condition in our hands, we replaced the stable version of HB60 with the wild-type HB60, and we successfully crystallized and solved the structure of the wild-type football with ADP and beryllium fluoride. In general, the structure of the wild-type football resembles that of the previously described HB60 mutant, which was published by our lab and in 2015. So the cycle, like by now, from our structural studies, is like the ATP trigger the formation of the double ring HB60. After that, HB10, the helper protein of HB60, bind. After the hydrolysis of ATP, the 2 rings separate and split to form the half football, and just later, the ATP and HB10 released. Thanks.
Hi, my name is Guy Lebel, and I study, I'm from Tel Aviv University, and I study ion channels, most specifically the Kv72 and Kv73. These channels are very important for the regulation of neuronal activity, and these channels are stop the action potential in neuron, and the PIP2 is a membrane, is like a membrane part, and it's activate the Kv72 and Kv73, and it's part of the membrane. And my what? This is from our lab, yeah. This one, yes. And this is another structure, but this is a model of the PIP, okay? And my project is to co-crystallize this activator, this PIP2, with the part of the ion channels, or the C-terminus part, and to analyze the binding site of them. Okay?
So there are 2 more students. Yes. Maybe they can introduce themselves.
Yeah, I'm smoking.
Okay, can you come here?
Hey, my name is Faris Salama, and I'm starting working in the crystallography center in Technion, and actually we're giving a service solving crystal structure, and that's it. I don't have my own project, so that's why. So.
Pierre Harris
He's not here. No, he's not here.
He's not
coming tomorrow.
Okay, so then we are through.
We should come back at 4 o'clock tonight. Come back at 4.
Yeah, we can come back at 4 o'clock.
15 minutes early.
Yes, yes. So we are ahead of schedule, which is quite surprising to me.
Sorry? Maybe there's no food. Maybe no food. The working details of what's going to be happening in the next few days. So inside the blue bag that we received, Eric is holding it up, are details of what she's going to present, so you can follow it that way as well. These details are extremely important because when we begin collecting data, as she will tell you, it's going to be a very tight and carefully controlled schedule. Orly.
I'm going to be.
Oh, I'm sorry. I'll get it for you. You can sit. I'll do it. I get it for you. I didn't realize we were going to do it now. One moment.
So first of all, I want to welcome everyone. Hopefully we're going to have an enjoyable workshop, and maybe we can also collect some data. Okay. All right. So, that was fast. Okay, so first I'm going to talk a little bit about the computer setup for the remote data collection and for the data processing. How are we going to connect the computers at the Weizmann Institute, also to do use some software during the workshop. How are we going to get the user ID, and we're going to divide you guys to 2 groups. Hopefully you looked through the notes. We're also going to go through the schedule for the ESRF and DLS data collection. The ESRF is on Tuesday, on Thursday is the DLS, and how we can connect and transfer files, MTZ and PDB only. Like we said, we don't want you to retrieve all the actual frames from your data collection, because otherwise we're going to be kicked out of the Weizmann Institute. So we want you to come back here, so we won't do it this time. And then some Linux command. Is there anyone who is not familiar with the Linux? Perfect. So, okay, I can one-on-one, we can go over it. Okay, so we don't have to go, so we don't have to go through that. Was it? Okay, so tomorrow, right, that's the first one? Yeah. So tomorrow we're gonna be in a different, as you understood, in a different place, in the Lupati conference room. All the lectures and the tutorial and data processing will be at the Lupati Conference Center. Just opposite it, the same place, we're gonna have data collection. At the data collection room, we're gonna have all the remote data will be done at this room, okay? All the students, even the one which did not send their own crystals, will have the opportunity to remotely collect data, either on a test case crystals if they didn't bring their own data crystals, or on their own crystals, both from ESRF and DLS. Again, I'm going to show you the schedule very soon. We're going to give you a username. In a minute, you'll see which one you got. You have it in your handouts. We'll try to do it. We have at the place where we are doing all the processing, we have 14 computers. You cannot use your own computer in order to process the data, okay? So, but we have 14 computers, which 2 of you will share one computer. And I guess because sometimes people will collect data and they will be in the Lupati in the collection room, they will not use these laptops. So sometimes you will have your own laptop, okay? What else? Also, you've probably realized that the event is broadcast, and you can find the site on the homepage. And so, like we said, in the data collection room in the Lupati Center, we're going to have 2 computers for data collection, okay, for the remote data collection. And we divided all of you to 2 groups, group 1 and group 2. And again, we're going to look at the schedule very soon. In the big hall, we're going to have, like I said, 14 computers for data processing and so on. And like I said, each 2 people will share one computer. We do not want to tell you which one. Just if you see a computer, you know, a free one, you can work. And if there is no free one, just join to one of the students and work together, okay? If you have 14 of the computers are, 16 actually, are PCs, right? And we have 2 Macs, and some of the Weizmann Institute people brought their own Mac. So we have different setups for remote data collection. If you have a PC, which is all the computers at the remote data room, and also at the Lupati conference room, there when you wanna connect to the PC, you double-click the mob extreme. You have it. Everything is set up on the computer, and then in order to log into the computer, you have to use your user ID. In a minute, we'll see what it is, and the same password for all people. Okay. Once you're there, you can actually create your own directory. So each person will have his own directory for his own protein or test case. And then you can go to your directory, and from there you can run all the software that we're gonna try in the workshop, okay? So we want to make sure that each one would have a separate directory for their own data, okay? Okay, if you do have a Mac, I mean, if you happen to work a Mac, we have 2 to give you, but other people which brought from the Weizmann might have a Mac. For that, you have to install VNC. The Mac that we give already have the VNC, and I asked from the Weizmann Institute participant to install the VNC. If you don't have, we'll help you tomorrow. And then, the way it works is as follows. Okay, let's do it that way. Okay, we'll go here. Opa. This is a very important slide. Okay, so if you're working on a PC, this is your user ID. Okay, it goes alphabetically, and that's student 1 until 29. That's all the students or the participants. Okay, if you need to use the Mac, then up to number 9 we're okay, because if it's either student 9 or Collider Weizmann ACIL 9. But for some reason, Kirill, the person who is in charge of the computer, could not get the number 10 and 11 to work. So here you have to, it's a little bit confusing, but everything is written. So you have to you don't have to memorize, just look at your name, and then you know which one you use, depending on if you're on a PC, on a Mac. Most of the people, we use that. So it should be straightforward. Okay. For the ESRF, we're going to collect data starting from 12 to midnight tomorrow. Okay, we're going to carry out the data collection ID 29. It's a tunable microbeam with a wavelength 6 to 20 kV. Here we show how you remotely connect via the browser to the ESRF. And tomorrow, Stephanie and Sasha will talk a little bit more about it. We're going to use MX-Cube3. When we want to download files from the ESRF, again, the PDB and the MTZ files, we can either use the ISP or SCP command. Again, you have here all the information. The MTZ, you can afterward, when you go back home, you can also download all the frames and everything. You can go back, as you understood, at ESRF, the frames will be there for a long time. Even if you decide to travel in Israel after the workshop, you will have time by the time you come back home. At DLS, again, the experiments will will be on IO4. And then again, it's a tunable beam with, that's the size of the beam. Okay, and we're going to use the No Machine software. Okay, and we're going to get more explanation tomorrow from Nick and Neil. Okay, again, we have instructions how to download the MTZ and the PDB files, and if you have larger data, then you can either bring a hard drive, which is not possible now because we are remotely from there, but for small quantity of files, you can also do the rsync. Okay, so this is the way the groups in your handout. There was a mistake. Okay, these two people, okay, Sharuti and Ramita are switched. Okay, so this is group 1 and this is group 2. The way we're going to work up is as follows. Okay. So we're going to start tomorrow at ESRF. We're going to start at 12 o'clock. Some people will skip part of lunch. Okay. So from 12 to 12:30, that's Hadass. She brought some crystals. And then after that, Michal, 7 more crystals. After that, after lunch, it's going to be from 5 to 3:30. We have these people working. Each one going to get allocated 15 minutes. They're going to work on test case. So it's going to be 1 crystal, basically. Okay. We're going to work during lunch, during coffee break, during dinner. You know, it's a hard time. You didn't come here to have fun. Hopefully with data, but you have to work for that. So that's going to be until 3:15. Okay, that's all the test case, and then we're going to go again with 2. That's already group 2. So while these people are collecting data, okay, in the small room, when one person is collecting data, all the people from group 1 will be there. The rest of the people, we have a different session, right? We have the dials session for group 2, which is going to be opposite the remote data collection in the center, okay? And then we're going to switch. When group 2 are going to start to collect data, group 1 is going to go and have the dials session, and then group 2 are going to collect data according to the schedule that we have here. Okay, by the time we get after dinner, I think, or at least almost after dinner, we have no sessions, you know, we have no actual sessions. We have here the schedule for people to collect data, and I guess the instructors are going to be available until when? Not until midnight, but until, I don't know. Okay, we'll see how awake they're going to be, but until some point, maybe after, I don't know if after dinner, but some people are going to still be around if you want their help in processing data and so on. Okay, so that's for the ESRF. For DLS, the same idea, okay? When one group is collecting data, even one person is collecting, all the group is together with them in the same room. The other group are still going to process data, either from ESRF or practice dials and CCP4 and so on and so forth. Okay, so it's the same idea. You have the schedule here. If you have questions, you are more than welcome to ask. I have to say that I'm going to be the bad guy because we're going to really make sure, like Rick did, he gave me a good example now. 4 minutes is 4 minutes. I won't put the stopper, but 15 minutes is 15 minutes, 30 minutes. So we'll, you know, make sure that everyone will be able at least to practice and hopefully to collect also data. Okay, we do have 2 data sets that we can work on. Okay, they're valuable in all the computers that you're going to log on. They're there, and we can practice on, you know, processing these data. I'm not going to go through these commands. Whoever needs help with the VI commands, then I can help them. If you have, yeah.
So just to make it clear, stay.
Yeah.
The 16 computers that are in the large conference room that you'll use, basically, although they're PCs, they're in essence just terminals. Yes. So I think there's no password even to get into them because there's nothing you're going to do with them. They're going to lead you to what we call a cloud computer called Collider here at the Weizmann that's set up just for this course with all the programs. So you just will log in, and so it doesn't matter which computer you're using. You will use your student name and this one password to get into the bigger computer, and the other one just the front end for that bigger computer. Okay, go back.
Your code is also working.
Yeah, everything is working.
Yeah.
Everything.
So even
all the list of all the programs.
Go over the list.
Yeah, we can go over the list. Okay, So all the, all the programs here should be working. COOT, CCP4i, I2, Phoenix, all this, okay? The app, the XTS app, PyMol, dials, everything should work in all the 16 computers, but 2 computers in the data collection room, you should not use them for any of these, only in the processing, in the other room where we process the data, where we have 14 PCs, 2 Mac. So if you have any other questions, and while we're working, you know, we're gonna be here and help you. Yeah.
Speak louder.
It is auto yeah. Right. Right. Thank you. Okay. So there is a sorry. If you don't want to get only your MTZ files, but maybe also your structure while you collect data, so I would ask you to send me first of all the sequence of your protein. If you remember the acronym of your protein, that would be easier. W, whatever it was, test case, whatever it is. And if you know a PDB of your either protein or a related PDB, please send it to me as well today, so we can enter all this information to DLS, ESRF, and then we can go even, you know, the results are going to be even more. We won't write for you the papers, but until the structure.
That's not just for the experimental phase, and that's if you're looking for a ligand or you
right.
try out you've got a whole load you think might be a decent target. If you want advanced answers, send that information through to our
Try to do it today so we can enter all the information to the book.
Other questions? If you have your own computers, then... Sorry, you don't know which?
Secure Wi-Fi.
If you don't have secure Wi-Fi, it won't work. To get into this cloud computer, it's actually inside the firewall of the Weizmann. So all of these computers that we're providing have that. Otherwise, everyone can use the guest one, which is Weiss Hotspot, but that won't get you to the local cloud computer. Yes? What? You can of course collect ED-ROM here, but you won't be able to get into the local cloud computer. That's inside the firewall, and only the ones which are blessed. We can't bless the ones which are coming in too quickly. Other question? All right, so I have Rick.
Maybe it's good to mention that if you start processing these dials, for example, you do that in as shown there, so that you start up these computings in a certain direction.
Yeah, that's why I said
yes,
before, right.
Exactly.
So I mean, we would like you, if possible, you know, like once you get into the computer, make your own a directory of your own project, and then whatever you run, just have something which will, you know, will know that it belongs to you and not to someone else.
The next day you can find it.
Well, that's also true.
So and if you have questions tomorrow, there'll be a lot of people around, and so there will be a small group that will be working, processing, another group inside collecting. So it won't be difficult to ask someone. And if you're not certain, just ask, better than to make crazy errors and waste your time. Other questions? All right, so I have one request. On Wednesday, we're not collecting data. It will not be relaxed, you'll work. But we're going to have a festive dinner in Tel Aviv. We're going to go by bus to Tel Aviv. It's a very nice restaurant looking at the Mediterranean Sea. But if anybody is not going to come, please tell me so we can get a better estimate to tell the restaurant, so they'll have enough chairs for everybody, okay? So if you don't plan to come, just send me email or just tell me in the next few hours, okay? So, yeah. Bus will come back to the Weizmann, yes. We're not going to leave you in Tel Aviv. I mean, if you want, you can stay overnight. It's a very nice city, but and there's a train you can take in the morning. Other questions? All right, so if anything is, I mean, I think Orly is presented very clearly, it's all written down, but if anything is unclear to yourself, don't be shy to ask us, even during the coffee breaks, it's okay. And I must say, it looks very simple now, but the organization that went into this was Orly and Christian and Rick did everything. Okay, great. Thank you very much. So coffee.